My name is Sharon Rounds, and I'm the Program Director for Advance RI Clinical and Translational Research Center. And it's my pleasure today to introduce our monthly Advance RI-CTR Research Seminar Series. And today we're really pleased to have presentations from two of our highly competitive pilot project program awardees. The first presentation will be from Dr. Ari Pelcovits, who is in the Hematology Oncology Division of the Department of Medicine and expert in the treatment of lymphomas. And the second presentation will be by Patricia Sullivan, who is Assistant Professor of Neurosurgery and who will be discussing her work with recurrent chordomas. I think in order to keep things moving, I would ask participants to put questions into the chat that we can address at the end of each presentation. And at this point, I'll turn things over to Ari, who will be discussing indolent lymphoma and the tumor microenvironment, novel insights from spatial transcriptomic analysis. Take it away.
Perfect. Thank you so much, Dr. Rounds, and thank you everyone for having me today. Like Dr. Rounds mentioned, I'm a hematologist and I focus most of my clinical time on lymphoma and other hematologic malignancies. Much of my research background so far has been clinical trials and outcomes research, but this grant has been a great opportunity to do some translational work and really work from bench to bedside, and I'll make note of my colleagues who've done a lot of the heavy lifting of this presentation at the end. So our objectives for today in the time we have together, I'll try to briefly review a little bit about indolent lymphomas, and in particular follicular and marginal zone lymphoma, which will be the cancers we'll be focusing on today. And I'll use that background review to bridge a little bit into this conversation about the tumor microenvironment and why we think it's important and of research interest. And then I'll get into the sort of data we've developed so far from our research using this technology called spatial transcriptomics and what it's telling us about the tumor microenvironment and research we're hoping to do in the future. So we can classify lymphomas in a lot of different ways. We can think of them clinically and how they behave. We have our indolent and aggressive lymphomas. We can classify them based on their cell of origin, B cell or T cell is the majority, with most of those being B cell lymphomas. We have these sort of more historical classifications that more people might be familiar with, called Hodgkin or non-Hodgkin. And then we might classify lymphomas based off of certain cell protein markers or other transcriptional markers that might have implications for prognosis or therapies. And today we're going to be focusing on lymphomas in adults, and in particular, these two lymphomas, marginal zone and follicular, which make up the significant minority of indolent lymphomas. These two lymphomas on our sort of classification system are indolent lymphomas. They're B cell in origin and are CD20 positive, as most B cell lymphomas are. But uniquely, or amongst a lot of indolent lymphomas, while they'll often be indolent, they'll have these slow disease courses where they'll not require treatment for many years. A small subset will become more aggressive, or after initial therapy will relapse quite quickly. And there's a lot of research into this space of trying to identify which of those cancers will not respond well, and how might the tumor microenvironment be playing a role. And so when we think about the tumor microenvironment, I think it's helpful to take a step back and just think about how do we diagnose lymphoma, and where does the tumor microenvironment play a role in that diagnosis, and why are we looking at it in particular when we're thinking about therapeutics. So like all cancers, lymphoma is diagnosed somewhat on clinical features, but significantly on pathologic findings, where sort of basic cellular microscopy is the mainstay of diagnosis. And sometimes morphology alone might be enough, but oftentimes we'll need certain unique staining methods, looking for specific cell surface markers or genetic abnormalities. And these cytogenetic and molecular and transcriptional studies that we perform are often more informative prognostically or predictively, and maybe less essential for diagnosis. And this is just a representation of what a pathologist might look at to make the diagnosis of follicular lymphoma. They might start with sort of a low-powered view of a lymph node dissection, and you can see here, and we'll come back to this a little bit, so I'll make note of it. The presence of these follicles, which are sort of standard for lymph nodes. It's sort of the B cell hub where most of the B cell activity takes place. But you'll sort of have to trust me, and I'll trust the pathologist that this general follicular overview here is abnormal. And then they'll look at it at higher power view. They'll do certain stainings, and ultimately putting this all together, they'll come up with a diagnosis. So where does sort of the tumor microenvironment come into all of this, and how does it parlay into our research? And as you can see on that prior slide, that follicle, that lymph node, is a hub of a lot of immune cell activity. And what we know is that some lymphomas will take over that entire lymph node, will entirely efface it. And those are typically our more aggressive lymph nodes. These are like our Burkitt lymphomas, or perhaps our diffuse large B-cell lymphomas, where on the biopsy specimen, we're mostly only seeing malignant B cells. Other lymphomas, in particular Hodgkin lymphoma, these blue cells would just represent the only tumor cells present, and the remainder of the lymph node architecture is really immune response. But a lot of our lymphomas, and in particular marginal zone and follicular lymphoma, live in this sort of middle space where there's this interplay. There's tumor growth, there's B cell malignant growth, but there's a lot happening between the B cells and the T cells and other immune cells in that microenvironment. And the way they're interacting may be important. And we know this also from our therapeutics. So chemotherapy has long been the backbone of treatment for lymphoma, but over the last several decades and really over the last several years, we've had advancements in the way we engage the immune system to attack lymphoma cells. So one of the first of these agents was one that I think more people are familiar with, which is rituximab, a monoclonal CD20 antibody. And this targets those B cells, that cell surface protein that is on many of our B cells. And there's some direct apoptotic mechanism of these medications, but they also allow the tumor microenvironment, those other immune cells, to engage with the cancer cell in some way they weren't before and attack and kill the cell. More recently, we've sort of expanded on the monoclonal antibody technique by creating these bispecific antibodies. So these retain that CD20 recognition, but they also contain another antibody which targets T cells directly. And by engaging T cells directly with this second antibody component, they're more effectively able to draw T cells into that lymph node lymphoma engagement and lead to cell death. And we're seeing a lot of exciting outcomes with these medications. Several of them have been approved in later lines therapies in lymphoma. And I'll mention that we have a clinical trial open, which is where our patient data come from, looking at this drug, mosunetuzumab, in the first line setting. So we know the tumor microenvironment plays a clinical role. We kind of see it cellularly on this microscopic level, but how might molecular and transcriptomic analysis tell us a little bit more about the tumor microenvironment? And what do we know up to date and the limitations in the science so far? So this is a study that looked back, back in 2004. So this is 20 years ago, and this looked at a population of patients with follicular lymphoma, about 200 patients almost. And up here on the right is a survival curve of all of those patients in this study. And they did whole genome sequencing on these patients, on these lymph node samples. They looked for sort of gene signatures, groups of genes being expressed that perhaps might be prognostic. And they did. They found these two unique gene signature clusters. So these two clusters of genes that seem to correlate with unique survival outcomes. And one of those, the favorable group, is sort of this grouped survival curves on the top, and the unfavorable group is on the bottom. What they weren't expecting was that these gene profiles were not profiles of B cells. They weren't representing something different about the malignant cells in the favorable or unfavorable, but they were actually representative of the T cells that were involved in these biopsies. So this was sort of the first sense that perhaps something else in that microenvironment, something else going on in those lymph nodes, is actually responsible prognostically, and that microenvironment is important. And this work was built on over the past several decades. And this is just one example of how more advanced sort of sequencing technology has been used. So this was a study from a handful of years ago looking at single cell RNA sequencing. So that prior study was whole genome sequencing on the tissue as a whole. Now this was when we were able to do single cell RNA sequencing. This was actually only of 6 patients with follicular lymphoma, but the RNA sequencing on the single cells was able to give data on 20,000 cells. And they found some expected findings about differences in B cell clonal populations between samples. But again, what was more interesting is this, as on the graph, is a heat map of T cell genes expressed. And what they did is they compared those 6 follicular lymphoma samples and they compared it to healthy lymph node samples, both from those patients themselves. They used tonsillar tissue, but also other specimens. And they found that the T cell genetic expression on an RNA single cell level was different in those healthy lymph node specimens compared to the lymphoma specimens. So further evidence that the tumor microenvironment and the surrounding T cell processes are probably playing a role prognostically and perhaps therapeutically. The limitation of these studies is that both of these techniques require degradation of the whole tissue. So the tissue architecture is lost when you run these studies. And while you're able to make inferences about these cells and perhaps how they're interacting on a microscopic level, you don't really know what's happening spatially in these tissues and where these T cells, where these B cells are, and how geographically their relationship might play a role. So that's what our data has been looking at, which is how can we combine this spatial data from our sort of just basic cellular techniques, microscopic techniques, with this molecular data? And can we learn something more about the tumor microenvironment by spatially locating this sort of genetic panels that we're obtaining? So our study used something called spatial transcriptomics, but in particular the Visium 10X platform. There's several different platforms that exist to try to do this work. But this is the Visium 10X platform. And the way it works is they provide these unique slides that your tissues are placed on. So they're slides kind of just like any other slide that we use, but they have these unique capture areas. And each of these capture areas have these, tiny microscopic wells with barcoded spots. And so what happens is that the cells are lysed and the RNA is released in each of these wells. And that's where that RNA sequencing is able to be performed, except where that tagging, that barcoding tagging, is usually just identifying specific cells. It's able to sort of geolocate it back to these wells where they were obtained from, because each barcode will match directly to one of these wells. And so once you obtain your sort of your readout of your RNA sequencing, you can map it back onto your original tissue. And so that allows you to both get the RNA data, but also retain sort of spatial awareness with your tissue. So this is just a layout of the studies we've performed so far. So we've selected 10 patients from our clinical trial. And as I mentioned, these are patients with follicular and marginal zone lymphoma receiving this unique bispecific antibody, mosunetuzumab, in the first-line setting. This is not currently an FDA-approved therapy. It's a clinical trial we have open. This medicine's approved in the later line, and we're one of a few centers in the country that have been running a trial like this, and we've been seeing some really exciting outcomes. And our hope in this trial was to do a few things. One is this technology is new, and there really just isn't a lot yet that's been reported about spatial transcriptomics and these lymphomas. There's minimal data about follicular lymphoma, and there's no data about marginal zone lymphoma. So this is really, on some level, an exploratory analysis with this new technology. The second part, which we're in the process of doing now, but I don't have the data yet to share, is looking at biopsies of patients on treatment. So patients both pre-treatment and post-treatment, in particular patients who don't have the responses we're hoping to see, and to try to get a sense of, is there something happening in that tumor microenvironment that's not exactly what we would hope, that could tell us something about the way we should augment their treatment. And so we're going to go through these different analyses we've performed and the data we have so far. So the first is really just the basics of what spatial transcriptomics is. And so I'll sort of show how this technology is run and how you then develop these mapping systems to place the data back over your initial tissue. So when you first run this data, it takes a lot of legwork. So the system has some statistical software, and it will look at all your RNA sequencing data, and it will look for what we call sort of spatial regions or spatial clusters. And it will try to group each of your RNA data points into spatially oriented clusters that share some similarities. Now, what the top row is sort of demonstrating, this is not a spatial representation. This is just a graphical representation of these different cellular regions based off of RNA signature, is that there's a lot of background noise. So each of these samples is going to be different because of perhaps what was sampled in each patient and the amount of B cells and T cells it contains. And so your first readout ends up just sort of splitting each of these clusters, each of these regions out as just each sample you ran through the system. So you end up with one spatial area per sample, which is not really helpful. So we're able to use some sort of other mathematical advanced systems to try to silence out that background noise, to say, okay, we recognize that there's going to be between sample differences. Let's try to really find the spatial related areas that we're seeing across samples. And that's what the sort of bottom graphs are showing. And ultimately, we've identified 4 spatial regions of interest, or spatial clusters of interest, that are shared between each sample. And what we mean by that is that there are going to be regions of space in the microenvironment on each sample where certain RNA signature groups are shared. And we'll show you what that looks like on a heat map and with some cellular data later. And we sort of identified these, and you'll see kind of how we came up with these names in a moment, as the germinal center sort of cluster, the border zone, the interfollicular zone, and the stromal zone. And what's nice about what I'm going to present next is there's sort of some internal checks that, is this just more noise you're seeing, or do these really match with geographical regions on these lymph node biopsies that make sense? And so this is what this sort of first spatial overlay looks like. So we take those spatial cluster regions that we've identified, and we apply them sample by sample to see if they're kind of matching up with regions that we would expect to see. And this does, it overlays nicely with what we would maybe expect going into this study. We're seeing these follicular regions, these follicle or germ cell regions, germinal center regions that I mentioned on our initial biopsy. And as we'll see, we're going to see some cellular and RNA data that really fits with what we would expect to see in those regions. What is interesting, and what is maybe hard to tell just on normal microscopy, is that what's outside those follicles, there seems to be unique clustered zones. So there's this unique border region that surrounds each follicle, which is different than these interfollicular regions, this other space that's spreading throughout the lymph node sample. And then there's going to be some sort of stromal regions in the background, which actually may be more important than we first gave credit to, but we're not going to get into so much today. And so over here is a follicular lymphoma sample, and this is a marginal zone lymphoma sample. And like I said, we're looking at differences as well between follicular and marginal zone. I won't get into that in too much detail today, but you maybe can tell a little bit these sort of follicle centers are less well defined in the marginal zone, which we know from just regular microscopy. So that's what it looks like, but what sort of information can we derive from that? Okay, so we know there are different spatial clusters, different zones, different cellular zones on these samples. Perhaps that's not too surprising, but what type of cells are in each location? Is there cells of interest that exist in one cluster and not another that we might be able to utilize from a predictive or prognostic level? And so the way we did this is we took that RNA sequencing data, and then we converted it back into information about cell types. Now, if you remember, we described the way this platform works as using these wells, these areas to derive that RNA sequencing data from. Each of those wells, though, is actually larger than just a single cell. So in a way, this isn't truly single cell RNA sequencing. So that data needs to be deconvoluted. We need to take the genetic data we get from each well and find a way to then interpret and pull out what cells that might be made up of. And so what we use is an RNA database of genetic markers that match with certain cell types to then sort of well by well deconvolute that data and determine what type of cells are probably made up in each region. And so what we're able to find is that there are unique cellular groupings within each region, which is what we would expect. And we're getting a little bit more information, though, about some interesting findings about what type of cells exist in each of these sort of tumor microenvironment regions. On the left-hand side here, it's a little bit busier, but this is each of the samples and what their zones look like amongst different cell types. But on the right here is looking at these clusters in general across all samples. And so again, this is a little bit of some quality control. This germinal center zone is consisted heavily of B cells, which fits with what we would expect. Where things got a little bit more interesting is looking at the differences between this border zone and the interfollicular zone and the germinal center zone. And what we see is that the T cell involvement in that border area seems to be increased compared to the other clusters. So that there's something unique about what's happening with the T cells in that location that's not happening in other locations. And we do see that this is statistically significant. This isn't just, appear to be this way, but we do see a statistically significant overrepresentation of certain types of T cells and other immune cells in that border zone compared to the other zones. And again, as we would expect, increased B cells in that germinal center. So it seems that the border zone is playing a crucial role in T cell activity. And again, this is just more sort of statistical analysis looking at overrepresentation of certain T cell types in each cluster, and again, showing the border zone as a hub for T cell activity. And again, interestingly, that interfollicular zone actually has fewer T cells comparatively, which is something we wouldn't have been able to necessarily recognize from initial just sort of general microscopy. So we've identified sort of our general way this system works. We've found unique cells in each of these clusters. But what does the raw RNA data tell us? So perhaps we can learn something from just sort of looking at this differential gene expression about the genes that are overexpressed in each of these clusters. And on top of that, we were able to pull from other databases these gene sets that might be important. So if you remember, that initial paper showed that certain gene sets might be indicative of unique immune environments. And so we do know from these databases that certain gene sets are indicative of certain cellular activity or cellular pathways. So we were able to see, are certain gene sets overexpressed in each of these clusters. So first, this is just a sort of a heat map of the RNA data, which again is as we expect. There are unique genetic expression in each of these clusters, and that's how these clusters were initially identified, and that this expression is statistically significant differently in each of these clusters. But this is our gene set analysis. So again, looking at a database that has gene sets that are indicative of certain cellular pathways and cellular mechanisms. And I'll point your attention to the right here. This is the border zone again, and looking at which gene sets are overexpressed in this area. And again, it's sort of matching with our other findings and adding to that T cell activation, T cell differentiation, and cell-mediated immunity seem to be really active in this unique border zone. So sort of the last thing we're looking at, or not the last thing, but one of the last things we're looking at is we also have database sets of cell-cell interactions. So what we've looked at so far has told us about the cells there and their activity. But what we're probably even more interested in is how are those cells interacting? How do those T cells engage each other? How do they engage the B cells? And that's something that we're continuing to look into, and we're seeing some promising results from. But just given time constraints, I wasn't going to present today. And then not pictured here, but as I had mentioned, we have a few more samples we're running now, hoping that we can really confirm some of the differences we're seeing between follicular and marginal zone lymphoma, that we can solidify some of these findings that I've presented so far. And lastly, that we can really look at these pre and post-treatment samples to see how does the tumor microenvironment change on treatment. I think what we've seen is that the tumor microenvironment plays a critical role in the development and prognosis of follicular lymphoma, that these advances in spatial transcriptomics are giving us a unique way to understand the tumor microenvironment, and I think we're hopefully moved towards a time where that can provide prognostic and therapeutic information. Our data is showing that there are these unique cellular spatial domains within these indolent lymphomas that have unique cellular makeup and cell-cell interactions. And in particular, I think where our focus is on this border zone, which appears to be a hub for T-cell recruitment and activity. And so I just wanted to give acknowledgement and thanks to people who really did a lot of the hard work behind this. Again, I'm mostly a clinician and a clinician researcher, and so this grant has been a great opportunity to really spread that out and participate and work with people across our department. So we worked closely with the Dubliecka lab, with Anna Trzalska, who is a co-PI on this. Michaela Pardo is a PhD student who really ran a lot of the Visium technical analysis. And then Adam Olszewski is my mentor and a clinician, and did a lot of the heavy lifting with the statistical and mathematical analysis. So thank you all, and I'll take any questions.
Okay, great. Well, thank you so much, Ari. That was really terrific. It's the technology is phenomenal. For somebody like myself who grew up looking at histology specimens only and drawing so many inferences and not having really a clue what was going on, this is just remarkable. So I have kind of a naive question. What if you took a normal lymph node from a patient with one of these lymphomas and did the same type of analysis on their normal lymph nodes? Would you see the same patterns in the different zones that are not necessarily malignant cells? And the thing I'm getting at here is there's something about the host, that environment, that might be somehow affecting the progression of or the initiation of the malignancy?
Yeah, no, I think that's a great question. I think the answer is probably yes and no. So there's one other paper out now that has done some spatial transcriptomic analysis of healthy normal lymph node tissue to try to, again, create almost like a biobank of normal so we can compare it to.
Yeah.
And their focus was a lot on sort of stromal cells. So what's going on in that sort of background, what we would have thought was sort of unimportant structural architecture. And they think that there's a lot happening there that's important. I think you're right that there's probably some of this activity already present that then the lymphoma is triggering and changing. But I think we are sort of the first to sort of describe this particular area and this T cell activity that's happening there. But I think that would potentially be an important next step comparison to healthy tissue.
Well, it's very interesting and truly space age. So I don't see any other questions in the chat. So I think we'll move on to
Thank you so much,
Dr. Sullivan, and thank you very, very much. So our next speaker is Dr. Patricia Sullivan. Patty is a neurosurgeon, and she has been working with this very frustratingly difficult tumor, spinal chordomas, and she'll be telling us about her work exploring the heterogeneity of primary versus recurrent chordomas. So take it away, Patty.
Great. So I'm going to share my screen. Perfect. So I'm going to talk today about chordoma and sort of what we've been doing in the lab with the generous funding from the Advance CTR group. Can everyone hear me okay? I hope so.
Yeah, I can hear you great.
Thanks. And I'm in the OR, I'm just in between cases, so I apologize if you hear noises. Everything is blurred, so we're HIPAA compliant. But as a neurosurgeon, you can imagine things get a little crazy from time to time. So the work that I'm going to be presenting today really kind of borrows heavily from the work of a lot of the students in my lab. So I will sort of draw attention to them at the end, specifically Jonathan Arditi, who's one of our students who put together kind of a lot of these slides. Let's see. Okay, so chordoma is not something that most people think about or experience in their lifetime. It's a slow-growing tumor, and it's relatively rare. The tumors occur along the spinal axis, and the belief is that they originate from these notochordal rest cells. So these cells that are sort of living in the disc spaces, potentially kind of within the nucleus pulposus of the disc itself. These notochordal rest cells are part of sort of midline patterning development. So it's something that we all sort of have lurking, and it's been unclear exactly when the switch flips to create these kind of slow growing, but pretty significant morbidity tumors. Even when we do an on-block resection, which in surgical speak means we're removing the tumor with a clear margin, we still see extremely high recurrence rates, and it's local recurrence. So the presumption is that there must be some cellular sort of signature left behind, some cells that maybe have migrated beyond the what we presume to be a clear margin. And we don't really have a great idea why. And the molecular profiles of primary and recurrent chordoma are currently poorly studied. One of the reasons why is when you have a rare tumor, even when the tumor itself is quite large, it's hard to obtain enough specimens, especially enough specimens from within specific categories of patients to really get a good idea about molecular heterogeneity. So in our lab, what we sort of want to look at is the differential expression between primary and recurrent chordoma. We wanted to look at what genes are involved, what are the biological foundations for the recurrent phenotype. Specifically, is there some sort of matrix remodeling protein that's involved, or tumor microenvironment, sort of immune surveillance markers that would be involved, and what are the candidate targets for biomarker establishment for clinical intervention. One of the things that has been done previously, there's been a lot of work on brachyury, which is thought to be a transcription factor that regulates its own transcription, so it sort of drives its own transcription. when there were clinical trials sort of looking at knockdown of that specific gene, we actually didn't see that play out in any type of a survival improvement. We've also looked at tyrosine kinase inhibitors. When I say we, it's the collective medical we. There's really been no improvement in survival or local recurrence for patients with chordoma, even with kind of targeting what we think should be, should be a useful target. And that's what sort of has led us to be a little bit more interested in the heterogeneity between groups. So one thing that's really unique about Brown is that Dr. Diego Coslin is here. He's the chairman of neurosurgery. He's one of the world leaders in sacral chordoma resection. So we have quite a rich patient specimen bank here. In addition to that, Maggie, who's on the call, and I have been fostering relationships with the Chordoma Foundation to try to just pool tissue together. So just to kind of give you a sense of the literature, I know that obviously the lymphoma groups have much larger patient groups than we do, and are doing incredible work, but in the chordoma space, just with the rarity of this tumor, anywhere from 5 to 10 patients tends to be what's reported in our literature. Our hope is to kind of, increase that with community relationships and pooling samples, but it can still be sort of meaningful with a small number of samples. So we worked with 7 patient-derived sacral chordoma tumors. We had 4 primary tumors through recurrent. And just to clarify, if a patient has a tumor that is first diagnosed and biopsied, but has not been fully resected, that's considered a primary tumor. A recurrent tumor is a tumor that in almost all cases has undergone a complete surgical resection and radiation treatment. So very unique kind of populations there. We focus on sacral chordomas specifically because although the tumors occur within the spinal axis, we really did want to try to isolate one specific subtype. We utilized the nanostring encounter panel. The reason why is we were working with archived specimens. So before I joined the faculty, a lot of the tumor tissue was just being archived in the pathology department. So we didn't have a robust way of sort of sampling all of that fresh frozen tissue. We did thankfully see some nice tissue recovery and RNA recovery working with that FFPE. We looked at the differential gene expression and the KEG signaling pathway enrichment kind of within the nanostring encounter. For people who aren't familiar with nanostring, it allows us to utilize a more restricted set of genes. So instead of forming a full RNA library, we're looking at a more restricted sample set. So for RNA that's degraded, it's a little bit more efficient and you just get higher yields because you're not trying to establish a full library. So we looked at 770 genes. This is, again, genes that were selected specifically targeting pan-cancer progression. So genes that were isolated in other tumor types to play a role in cancer progression. 236 of the genes that we sampled within this pre-selected panel had no measurable expression change, and 56 genes had a statistically significant difference. So we saw 14 genes that were upregulated in recurrent versus primary, and 38 that were downregulated. And this is just sort of a graph sort of displaying what we saw with our threshold of statistical significance for those differential regulated genes. When we looked at pathway enrichment, it's actually kind of interesting. So the signaling pathways that are altered in recurrent chordoma specifically. So again, this is a tumor that has occurred, undergone surgical resection and radiation in almost all cases. We see the immune signaling and evasion, specifically TNF and TGF pathways, and chemokine signaling pathways are particularly altered. Growth and proliferation, which isn't terribly interesting, well, not interesting, but I don't know if that's necessarily something that's as helpful to us. And then you can sort of see the enrichment terms becoming a little bit less meaningful as we sort of move down to the bone matrix remodeling section. So we're kind of interested in that. Again, I think the immune signal and evasion, kind of backpacking off of Ari's work, I think that that's where a lot of scientists are looking now. One of the things that we find so interesting in recurrent versus primary chordoma is, again, in our samples, in almost all sample sets, recurrent chordoma has undergone radiation changes. So for a lot of these, local tumor areas, the immune system is really altered just from the effects of the radiation itself. I'm sorry. Okay. So the next thing that we looked at was RNA-seq. We were able to obtain tissue from 3 primary chordomas and 2 recurrent chordomas. Again, this was leveraging kind of our own tissue as well as tissue from the Chordoma Foundation. In this case, when we were using our RNA-seq, we were looking at total RNA. So this was with fresh tissue. We were able to develop full libraries. We used the GeneWiz RNA sequencing and then looked at kind of a literature review for each of those genes to see what was born out of the full library. So this is a reflection of the differentially expressed genes between primary and recurrent for our RNA-seq data set. So this was looking at a much larger set of samples, a much larger set of genes and a much smaller set of samples. We did see 60 genes with differential expression. You can see these here with reflected with the red dots. So 8 were downregulated in recurrent versus primary and 52 were upregulated. So one thing that's particularly interesting for us, chitinase 3-like-1 is a protein that plays a role in sort of immune evasion and tumor microenvironment. It modulates, we believe, the PD-1, PD-L1 axis, and that's something that was kind of exciting for us as a lab because without revealing too much of what is going on in the laboratory, this was a molecule that we had selected sort of for other reasons. So this was sort of exciting that we saw this differential expression in both our RNA-seq data, and if we go backwards, actually, you can see it in the RNA, the nanostring data as well. So this is a little bit of a messy figure, but one of the biggest questions that we had and the sort of bulk of why this figure is so exciting for us as a laboratory is this is a reflection of kind of the cluster analysis when we looked at primary versus recurrent tumors. And so the primary tumors sorted sort of separately from the recurrent tumors. Again, this is a small sample size, so I don't want to get too excited. But what it shows us is that it does appear that the primary tumors tend to have more of a homogeneous profile compared to those recurrents. And even the recurrent tumors just show such disparate sort of clustering even amongst those 2 samples. Again, it's very important that we take this with a grain of salt, but you'll see the chitinase 3-like-1 is sort of, I'm not sure if you can even see it because it's probably so tiny on your screen, but that's one of the genes that was born out in this cluster analysis as well. So we did a little bit more sort of digging at how these genes were clustering specifically in our first recurrent tumor. And the other thing that I will say, we're kind of gaining some sophistication in how we're looking at the heterogeneity of these samples. With our department and with our own internal sort of fresh tissue samples, we are able to get a much better demographic history historically that hasn't been reported. So the tissue that we were getting from the Chordoma Foundation, we're presuming that it was radiated just based on clinical practice guidelines, but it has been a little bit different. It has been a little bit challenging kind of obtaining really consistent reporting on the demographic analysis. Part of the other issue is that for a lot of these patients who are undergoing surgical excision and subsequent radiation, they're traveling all over the country. So people will travel here for a surgery. They might go up to MGH for proton therapy. They might try to travel closer to home. Some patients are also interested in exploring some of the carbon ion therapies in Germany or Japan. So a lot of these patients can be very sophisticated in kind of how they're seeking out care. So in terms of kind of looking at the demographics more closely, we're kind of working on standardizing reporting on an international level through an AO spine organization. It's just kind of a caveat here. Again, the clustering sort of recurrent 1 versus recurrent 2 was unique. We did see downregulation of chitinase 3-like-1 sort of in the recurrent population, which again this is just another way of looking at the gene data or the RNA-seq data that was on the previous screen. So this is sort of summarizing the patterns that we saw in recurrent chordoma gene expression, and this is born out of the RNA analysis, and that was cross-referenced with our literature review. So we did see sort of differential expression and metabolism, control, growth and proliferation, and again, immune signaling and evasion. Although from a practical perspective, I was sort of expecting to see more in the adhesion and bone matrix family. I think that it is interesting to think about sort of cancer targets that, you know, affect metabolism and growth and proliferation. So I think there's a lot to be done here in next steps. So when we think about how do we put this all together, we know that now, or at least from our preliminary data, We believe that recurrent chordomas are distinguished from primary chordomas by a more aggressive growth profile, migration beyond the bone matrix anatomic niche. And this is particularly relevant because we do see patients have these large tumors that kind of grow into the rectum, into adjacent neural structures, and even posteriorly into the spinal canal. So they do seem to not respect those tissue planes. Metabolic changes favoring greedy energy use, more effective immune evasion. Again, sort of we suspect that chitinase 3 like 1 plays a role in that. And these phenotypic distinctions are reflected in gene expression. Our hope is that this genome variant information is going to help us reveal underlying mechanisms and allow us to identify new therapeutic targets. One thing I will say is that for our next steps, we really are looking more specifically at radiation treatment effects. It's a little challenging because, again, for patients who undergo chordoma resection, sometimes those patients are going to be exposed to photon therapy, so high-dose photon therapy, although it's rare. Most patients, I would say, are going to be experiencing a pretty standardized radiation protocol with proton radiation. Yes. And then carbon ion is a new kind of emerging therapy. Another thing that's interesting as we build sort of our relationships with other institutions is while we have, one of the world's greatest surgeons here doing on-block sacral chordoma resection, you can imagine it's a highly morbid surgery. So a lot of centers, especially Memorial Sloan Kettering and some other centers that have the proton beam therapy, are really trying to kind of push the envelope as to what can be done with proton therapy alone to sort of spare patients from the from the morbidity of surgery itself. So those are two things that we want to look at kind of on a cellular level, because I think that what we don't want to do is make a distinction that this is intrinsic to the tumor itself and not take into account those radiation effects. And that's sort of our next steps. We're very fortunate in the lab to have a lot of really passionate students. So we have assigned several of our students to do a more in-depth literature review of all of those up and down regulated genes. And our next steps are going to be to kind of expand. We've, in the years since we've started the pilot project, we have acquired quite a robust human tissue sample. Actually, just this week, we're collecting another chordoma. And I think that, sending our own frozen tissue that we've collected here with the robust demographic data, I think is going to be a really good next step. Another exciting next step is that AO Spine International has taken an interest in our biobanking project here. One of the things that we do uniquely about biobanking, and Ari, this again sort of plays into what I think you were talking about, was we'd like to sort of look at how the tumors are blocked. So unlike in cases of lymphoma, where, again, if my memory serves me right from basic anatomy, the lymph nodes are small and can be plated on a single slide. Some of our tumors are 6 centimeters in size or greater. And so it's blocked on multiple cassettes. So we really want to kind of standardize how our pathology department is blocking, and specifically looking at, primary and recurrent tumors, comparing margin to margin versus core to core. So that, again, we're not it's more of the gross spatial transcriptomics of saying sort of in a larger tumor, are we looking at a hypoxic core? And is that why we're seeing more hypoxia versus a metabolically active margin? So I think I'm a little ahead of time, but that's all I have. I don't know if people have questions or if I should stop sharing.
Oh, that's great. No, thank you very much, Patty. It's so great to see all the progress you're making on this project, which I had the pleasure of learning more about a year or so ago. But this is terrific. So it seems like with a rare tumor like this, really a huge issue is the tissue banking. Did I hear you mention there's an international tissue bank for these, and how well phenotyped are those patients?
Poorly. The nice thing is that I think it's such a small community of surgeons that are all very well meaning. It's just, I think what we need to do is standardize how we're doing our tissue banking, because historically, these patients have recurrences over a 5 to 10 year period. So we're people have been banking tissue that started 5-10 years ago before a lot of this more sophisticated analysis was available. So I actually have a meeting on the 20th of this month with AO International to kind of propose how we've been doing our banking and recommending that kind of to the larger group. So whenever we take a specimen from the OR, we culture a cell line, we save a frozen sample, and then a portion of the sample goes directly to pathology. So when we collect the sample from the OR, again, part of this is just that our pathology department's been so wonderful and collaborative. When we collect that sample from the OR, we mark if it's ventral margin, if there's a hypoxic core. And so when we bank our tissue, we bank it as specifically as possible. Because I suspect that even though it looks pretty homogeneous on a pathological level, what we don't want to do is try to compare in these really tiny sample sizes, core and margin. Because again, I think that might be why we're seeing some of that hypoxia play out. That pathway kind of seems to be relevant where I can't find a clear reason why that would be super relevant, just thinking about the chordoma tumor type itself.
And how will you have some kind of a de-identified database for clinical information?
Yeah, so I don't know if you've heard about Flywheel. Neurosurgery has this big Flywheel database that's has an option for a firewall. So you can kind of create a wall between identifiable and de-identifiable data. One thing that's tricky is DNA is obviously considered identifiable. RNA, we're erring on the side of calling that HIPAA protected information because one day they might be able to trace that back. So we're working kind of in real time to define protocols and erring on the side of patient safety. So there's a question about what can be shared and what should be shared. And I think that as an institution, we're erring on the side of being more cautious. So sharing kind of RNA sequencing profiles, the nanostring, I think it would be very hard to argue that restricted data set would be identifiable, whereas an RNA-seq library, I think, would. And as far as the demographic data, it's really how long did the patient survive, male or female, radiation exposure, and just making sure that it's traceable.
Yeah. And Ari has a comment in the chat. Tissue banking is something we need to work on in the malignant heme space. We have a great bone marrow biobank as we have a relationship with the Dubieleka lab. But this could be improved with lymphomas as well. And I guess with all tissue banking this is something as a medical school, it would be nice to have a more unified approach to, but patient confidentiality, of course, is a huge issue, especially if you're sharing across institutions.
Yeah, the other thing that we do try to do, and I don't want to interrupt you, and Ari, I don't know if this is helpful, but we sort of sent out feelers through all of our brain and spine researchers to just ask what would be your ideal database? What would be the perfect database that you could use 5-10 years from now? And one of the things we also do is blood collection. So we use like the RNA stabilizer tubes for blood collection, and we do try to collect PBMCs as well. So we're trying to kind of proactively collect those specimens and kind of store the serum, which again is something that we'd want to probably, I think everybody is going to want to do that, especially if you want to look at circulating biomarkers in the future.
Yeah, we sort of have a lot of discordant projects kind of running simultaneously, and I think you're right. I think kind of a centralized approach to identify what everyone wants, because we have similar projects, but it's sort of like we don't have a good central hub for it all.
And then it becomes, even just the practical matters of making sure that if you audit yourself, we run like an internal audit pretty regularly, and I meet with our IRB staff, just making sure that everything is done correctly. It's a little bit labor intensive, but it's important. And then now that what's coming up is how do we make sure that our freezers don't go down? So we have to now invest in equipment that will send out a wireless alert, but what if the wireless goes down? So it's kind of, it's one of those projects that is almost like a garden that just won't grow. It just requires like constant pruning and constant attention, which is important. But I think just until there's a champion for it, it's hard to make it work. And if we don't put good data in, we're not going to get good data out. So that's sort of how we feel at the lab.
Yeah, very interesting. Well, I'm sure the Legaretta Cancer Center also has a great interest in this area as well. But well, so great. Well, that's terrific. Let's see. I don't see anything else in the chat now. So I just want to thank both Patty and Ari for really interesting, stimulating presentations. You're both doing really exciting work and challenging areas, and we're really excited to be able, from Advance CTR's point of view, to be able to support your work. And we hope everyone will join us next month, I mean. We'll, at that time, Bill Van Nostrand, who is a neuroscientist at the University of Rhode Island, will be talking about his work on the pathogenesis of cerebral amyloid angiopathy. So thanks, everybody, and see you next month. Bye-bye.